My name's Lewan Parker. I'm from Deakin University, Melbourne, Australia, and I'll be your host today for today's virtual journal club. I'll start by saying good morning, afternoon, or good night, depending on where you're logging in from. It's morning here in Australia. So joining us today, we have Professor Jose Calbet and Dr. Martin-Rincon, and they are from the University of Las Palmas de Gran Canaria. And we'll be discussing their recently published paper titled Functional Reserve and Sex Differences During Exercise to Exhaustion, Revealed by Post-Exercise Ischemia and Repeated Supramaximal Exercise. So also joining the panel today is Associate Professor Severine Lamon, Dr. Kim Wei, and Dr. Hannah Thomas, and they are from Deakin University, Melbourne, Australia as well. So collectively, the panel have extensive experience in human physiology, including muscle adaptation and hemodynamic responses to exercise and exercise training. And importantly, they all share a similar interest in exploring and identifying sex-based differences in this particular field of research. There's a few reasons why I've chosen this particular paper for the virtual journal club today. One of the primary reasons was the way that the authors have taken an integrated approach to their research. So they've used numerous validated techniques to comprehensively explore functional reserve and to also look at the physiological basis underpinning performance fatigability in humans. The other factor that I really applaud of their research is their effort in exploring sex-based differences in human physiology, including functional reserve, recovery, and performance. This last point's really important because research has shown that there are sex-based differences in numerous physiological processes in humans. This includes the development of cardiovascular disease and heart failure, hypertension, thermoregulation, exercise substrate utilization and adaptation, and also topical today, performance fatigability. Yet only 63% of research in sport and exercise science has included males and female participants, with 31% of the research including or investigating males only, and then only 6% of research investigating females only. And this is recent research from Emma Cowley at Liverpool, John Moores University. So it's quite clear that we do need to improve our effort in researching and then getting a better understanding of female physiology. So I'll hand it over soon to our leader or our panelist discussion. So that's Professor Calbet and Dr. Martin-Rincon, who will take us through their paper, their methods, results, and interpretation. After this, Associate Professor Severine Lamon will discuss briefly ways in how we can improve our study design to gain a better and more thorough understanding of female physiology. And then afterwards, we can have a quick chat. We'll go through some questions, and then there's a little networking session that we'll send a link, Zoom link in the chat afterwards. So I'm going to hand it over now to, I think it's, who's presenting first? Is it Professor, no, I think it's Dr. Martin-Rincon. Are you presenting first? Yep.
Thank you for the introduction. With your permission, I will share my screen.
Yep.
I would like to thank Physiological Society for inviting us to this journal club. It is actually a pleasure to share our research recently published in the Journal of Physiology. And just to give you a little bit of background about myself, I'm a postdoctoral researcher at the University of Las Palmas de Gran Canaria, and I've been several years now studying fatigue mechanisms, particularly leading to task failure, continuing with the research line established by Professor Calbet more or less 10-15 years ago. Then I would like to start with the introduction of the paper. And as quickly seen in this lengthy title of the article, I would like to introduce the term functional reserve for those that might have not heard of this concept yet. And we define functional reserve as the capacity to produce power at the same level or higher than reached at exhaustion. And now I would like to introduce you to the scientific background that has led to coin this term of functional reserve. And this term primarily comes from an integrative study from a group whose first author was David Morales-Alamo. And this research aimed to determine the main mechanisms of task failure during whole body incremental exercise and to assess if there is any functional energy reserve remaining at exhaustion to produce power. We used total occlusion of the lower limbs with instantaneous application of ischemia at 300 millimeters of mercury at exhaustion using a rapid cuff inflator as depicted in the picture there on the right. In these experiments, we recruited 11 males, and they perform one of the sets of experiments. They perform a 10-second isokinetic control sprint in resting conditions, and then after 5 minutes of rest, they perform an incremental exercise to exhaustion, either in normoxia or severe acute hypoxia. And then right at exhaustion, we applied this instantaneous bilateral leg occlusion at exhaustion, in which we impeded muscle oxygenation and the synthesis of phosphocreatine for 10 or 60 seconds. And then right after that period, we reopened the circulation, and we asked the subjects to perform a new sprint, 10-second isokinetic sprint, for another 10 seconds. All this exercise was performed in the cycle ergometer, as you see from the picture. In another set of experiments, we did the same protocol, but instead of doing a bilateral occlusion, what we did after incremental exercise to exhaustion is to only occlude one leg, and instead of sprinting, what we did was to obtain a muscle biopsy at 10 seconds from the occluded leg and a bilateral biopsy from the occluded and non-occluded leg at 60 seconds. I show you also this protocol because this will give us important information, given that in this study, we established from these muscle biopsies that no resynthesis of phosphocreatine can be achieved during this ischemic recovery. In this protocol, in this figure right here, we see the tissue oxygenation index from the NIRS just at the end of the incremental test, in which we see here the tissue oxygenation index. And then when we apply instantaneous bilateral leg occlusion, we see this very fast drop of O2, in which that O2 that it was trapped with the occlusion and bound to myoglobin is consumed in 2-3 seconds to achieve this complete deoxygenation of the muscle during the ischemic recovery. And then when we release the cuff, we see how during the sprint there is some reoxygenation of the muscle. While if we do the same, if we see the same graph with free circulation, we see how oxygenation is recovered during the free circulation recovery time. When we look at sprint performance, this is the instantaneous peak power obtained in resting conditions. But if we look at the sprint after incremental exercise in either normoxia or hypoxia conditions, this is the peak power that we obtain, around 600 watts in hypoxia, and this is the peak power obtained in the sprint performed after incremental exercise in normoxia. So we can see that this power output is much higher than the power output that we attained at exhaustion. And thus, this investigation showed that at the end of this whole body incremental exercise to exhaustion, a large functional reserve remains in the muscle to generate power, which reaches levels that are far above the power output reached at task failure. Studies in humans have shown differences in fatigability between females and males that are primarily intensity and task dependent. In these two figures on the left, we can see a task using the single limb knee extensor exercise with intermittent isometric contractions at low intensity, 30% of maximal voluntary contraction during 30 minutes. And we see how the maximal voluntary contraction is more reduced in these black circles for males than for females. And then here at the bottom, we see that the root mean square EMG, which is indicative of muscle activation, was higher for males during this protocol of 30 minutes. And then if we go to this higher intensity, 50% of the maximal voluntary contraction, which led the subjects to exhaustion, we see that the females were able to exercise for almost 3 times that time spent by the males. So the males fatigue much earlier at that intensity. And that occur also despite this shorter duration in males. We can see in the root mean square ENG that the muscle activation was even higher for males despite the shorter duration. And even when subjects were matched by force, the differences were still evident. And this study demonstrated sex differences in fatigability for intermittent isometric knee extensor exercise at low and moderate intensities. And also, overall, similar results have been described for muscles of the upper extremities with isometric tasks performed at intensities lower than 80% of maximal voluntary contraction force. In this study, they utilized electrical evoked contractions. And we can see in males and females, and we can see the decrease in torque with different intervals of contraction relaxation periods. And we can see how these black circles, which represent men, go farther down than they do for females. So that means that there is a lessened diminution of torque in the female group. And this arrow depicts where the differences became significant. And we can see that with these different intervals, in all of them, females seem to fatigue less than males. And also some other studies using transcranial magnetic and electrical stimulation have shown some differences in recovery of maximal torque after isometric and dynamic tasks that are performed at low intensity. And nonetheless, despite the evidence, I just try to outline whether males and females fatigue similarly during whole body exercise in the severe intensity domain remains unknown. And additionally, there is no evidence of sex differences in recovery intensity whole body exercise. There has also been suggested by some authors that the lower fatigability and faster recovery in women could be due to reduced metabolic impairment. And in line with this, recent work from our group has compared women to men with similar VO2 max, finding higher mitochondrial respiration when activating complex 1 and complex 1 plus complex 2, as well as higher protein leak and mitochondrial P50. And these differences can be important in the fatigue context by easing O2 extraction. Input from type 3 and 4 muscle afferents may also influence the rate of perceived exertion and affect also task failure via central mechanisms. And some studies have shown that men have a higher sympathetic and pressure response to metabolism reflex activation in males when compared to females. However, the majority of data come from males that are studying in a lying supine position and eliciting the metabolism reflex in the forearm muscles. Therefore, if males have higher metabolism reflex sensitivity than females, ischemia after exercise should result in a higher heart rate and ventilatory responses during ischemia in males. So here comes the main research question of a study, and that is, are there sex differences in task failure and functional reserve? Thus, the aim of the study was to determine whether males and females have a functional reserve for exhaustion following supramental exercise at 120% of VO2 max, and to ascertain what mechanisms may explain this potential between sex differences in task failure and functional reserve. We hypothesized that females would have a lower capacity than males, even when normalized to the lean mass of the lower extremities, that following high intensity exercise to exhaustion, females would have a lower functional reserve than males, that during repeated fatiguing high intensity exercise, females would achieve greater O2 extraction than males, and post-exercise ischemia would reveal higher metabolic reflex, induced heart rate, and ventilatory responses in males than females. In order to do this, we recruited 18 females and 18 males, all physically active, and we performed 2 different sessions in which they developed the following protocol. After a warm-up, the subjects performed this supramaximal constant intensity exercise at 120% of VO2 max until exhaustion, and then right at exhaustion, depending on the session, either in the ischemic session or the free recovery session, we applied in the ischemic session a bilateral occlusion using 300 millimeters of mercury, total occlusion of the circulation, or free circulation, allowing for recovery with unloaded pedaling during this period of 20 seconds. After those 20 seconds, in the case of the ischemic session, the cuff was released, and they performed a new bout with the same intensity until exhaustion. When they were exhausted, again, in the ischemic session, we occluded the circulation or allowed for free recovery with unloaded pedaling. And then after 20 seconds of either ischemic or free recovery, they got ready for this last bout of supramaximal constant intensity, again, 120% of the VO2 max. That was the main protocol performed in 2 different sessions, either the ischemic session or the free recovery session. Besides, we did some other measurements in which we assessed the VO2 max and the VO2 intensity relationship with an incremental test, which allowed us to calculate this 120% of VO2 max intensity for the constant intensity test. And we calculated the O2 demand during the supramaximal constant intensity test. We also calculated the accumulated oxygen deficit in the supramaximal test, which is an estimate of the energy provided by the substrate level phosphorylation. And that was calculated as O2 demand minus accumulated O2 consumption. And then from the O2 deficit, we calculated moles of ATP and lactate. And with a new methodological approach, we calculated the O2 depth incurred by the lower extremities during recovery. And that was done by subtracting the O2 depth during the ischemic session from the O2 depth during the free circulation session. We also measure during the protocol cerebral and muscle oxygenation over the frontal lobe using an optode measuring on the frontal lobe and vastus lateralis using NIRS, in which we use the tissue oxygenation index and also this TOI O2 extraction index that we calculated as the mean TOI during exercise minus the minimal TOI acquired during ischemic recovery. That's important to keep in mind for the results. And we also measured body composition using DEXA. Just to also highlight that in this study, we employed a full non-invasive methodological approach. And then now, before leaving the professor, Prof. Calbet, to start, I will show you a small part of one of our experiments, how is the setup and some of the procedures that we use during an actual test. There is no sound. Here we can see the subject starting the 120% supramaximal exercise. Now we can see the cuffs over both thighs, and here we can see how oxygen consumption is measured with the metabolic card. Here we see the integrated signals from the NIRS and also electrocardiogram. And the subject is approaching the end of the constant intensity exercise. It has been cut a little bit because it was too long for the video. He's approaching exhaustion. My colleagues are trying to push to make him push as hard as possible. And this is the recovery, where the calf was, both calves are inflated by this calf inflator that we see here, that immediately releases 300 millimeters of mercury to completely occlude the circulation. And then again, after 20 seconds, the subject starts again, resumes peraling, and he just put up the load, and then he's continuing with the second bout after those 20 seconds of ischemic recovery. And again, he got exhausted. There is again a total occlusion of the circulation. And then the 20 seconds are taking place. We are also asking for their opinion on the pain scale during this total occlusion. And again, this is the third bout after release of the calf. Okay, so that's 2 occlusions and 3 bouts of 120% intensity of VO2 max. And then from that point, I will leave Prof. Jose Calbet to continue with the results.
So, thank you very much for considering this paper for discussion in the Journal Club. So I will start just showing the characteristics of our subjects. So we recruited 18 males and 18 females. Females were slightly older than the males, but the difference was 3 years only. They were 23 years old, when the females were 26 years old. So the difference was significant, but it's very small, and we don't think from a physiological perspective it has any meaning. Then I would like to highlight that maximal heart rate was 190, 186, 89.6, so almost the same. VO2 max was 47 milliliters per kilo for the males, and the females 40 milliliters per kilo. The difference was 17% lower VO2 max per kilo body mass in females. And this difference is a little bit more that you will see in top level endurance athletes between males and females. That is very close to the difference that you will see in top elite athletes between males and females. Then if we did also the comparison for oxygen deficit among other variables, and in oxygen deficit, females, they had 22% lower oxygen deficit expressed per kilogram of lower leg of legs limb mass. So and the RER at exhaustion was 0.08 units lower in females than males. And finally, there was also significant difference in end-tidal CO2 at exhaustion, was a little bit lower, about 4 units, 4 millimeters of mercury lower in females than males. All the rest of the variables that we assess were comparable when adjusted for lower extremities limb mass. So the first point that I will address regarding results is the functional reserve. We define the functional reserve as the amount of work that can be done at exhaustion at the same level of intensity of exercise or higher. In the case of this experiment, subjects had to do exercise at 120% of VO2 max, and that's what we have here for the males and females, and this is the work they were able to do. And then when they were exhausted in one experiment, they had an occlusion of 20 seconds, and then they were requested to restart the exercise. And both males and females were able to restart the exercise. And you can see here that they were able to restart the exercise after the first bout, and they were able also to restart the exercise and produce some work after the second ischemia. That means that there was a functional reserve. There was a capacity to do exercise at the same level at the same intensity that produced exhaustion, despite that they were recovering with ischemia. In another experiment, the recovery was with free circulation, and they were also able to do exercise as expected, but the amount of work they were able to do was higher than when they were recovering with ischemia. But the amount of exercise was comparable in males and females. Here on the right, we have the main effects for the last two boats that are the most interesting here, because are the boats that are produced at exhaustion. And here we can see that the work performed by males and females expressed by kilogram of limb body mass of lower extremities limb mass was the same after occlusion in males and females, and also perfectly matched with when the recovery was also done with free circulation. So then overall, the amount of work that could be done after exhaustion was similar in males and females. So we conclude from this result that there is a functional reserve that is not exhausted after the first bout of high intensity exercise. So they can go further for another one. And actually, some subjects could do even more bouts of supramaximal exercise. And this functional reserve is similar in men and women when normalized for kilogram of limb mass. So then we look at the mechanisms that could explain this capacity to produce work after exhaustion. And we look at the oxygen deficit, and the oxygen deficit contracted after the first ischemia was also similar when normalized per kilogram of leg limb mass in males and females after the first bout, and a similar result was observed after the second bout, similar amount per kilogram of limb, lower extremity limb mass in males and females. And those values here, they were slightly lower than the value here, but the difference very small. The oxygen deficit contracted during exercise with free circulation recovery was actually of the same magnitude as the one observed after ischemia. So that means that the free recovery did not influence the capacity to utilize the anaerobic energy metabolism in the second and third bout when the recovery was performed with free circulation or ischemia. And here we have the mean values for the occlusion experiment and the open circulation experiment for oxygen deficit in males, blue, and females, red, and here in light blue and light red for males and females. And this is the overall mean value that is exactly the same when expressed by kilogram of lower extremities limb mass. In this slide, I'm showing the results for oxygen uptake. And the oxygen uptake during the first bout of supramax 120% VO2 max exercise intensity was similar in males and females. And after ischemia was also similar after the first ischemic bout, and after the second ischemic bout, it was also similar. So there was no differences between males and females in the amount of oxygen consumed per kilogram of limb body mass when they were doing exercise at 120% of VO2 max in the rested condition that we have here. And here we have the fatigue condition after ischemia. And in the right panel here, we have the same results, but with free circulation recovery. And you can see here that the VO2 is a little bit higher when there is a free circulation recovery compared to the situation where there is a recovery with ischemia. But there was no difference between sex, neither in the ischemic experiment nor in the open circulation experiment, and the overall mean values were similar. So this allows us to show that regarding the capacity to use oxygen in the rest and fatigue state, this capacity was similar in male and females in the rested condition or in the very fatigue condition. Then we look at oxygen extraction because, of course, we know that females, they have a lower hemoglobin concentration. And if we assume that blood flow per kilogram, blood flow is linearly related to the exercise intensity, and that the exercise intensity was comparable between male and females in relative terms, so it is very likely that blood flow was similar between males and females. So if we assume that leg blood flow was similar, so then the only way to explain that they were able to have a similar VO2 is by having a greater oxygen extraction capacity. Otherwise, females would have require a larger leg blood flow to match the oxygen delivery of males per kilogram of lower limb mass. So then the NIRST data indicated that females during the experiment with free circulation recovery, they were able to extract more oxygen than males. This conclusion is achieved by looking at the tissue oxygenation index during exercise and discounting the tissue oxygenation index observed at the end of the ischemia period. And this index, we validated this index in a different experiment with another set of subjects. And this index was correlated to the oxygen extraction capacity of the subjects studied. But this difference in oxygen extraction capacity was only observed with free circulation recovery, was not significant, and the difference was very small with in the experiment that was performed with ischemic recovery. So this slide is dealing with recovery of performance. So here what we have is the percentage of work capacity. We assign 100% level of 100% exercise capacity to the performance achieved in the first boat in the rested condition. And then what we have here is what is the amount of work that is performed in the second boat, what percentage represents of the 100% that they had in the first boat. And what we can see here is that females, we can go to the right panel, they have a slightly higher recovery of performance when they are doing the exercise with open circulation. The effect is not significant for the exercise performed after ischemia. Also, there is a small difference here that this didn't reach statistical significance. So that meant that in some circumstances, female may recover slightly faster or slightly more than male. Then we looked at metabolism during recovery, and we saw that the occlusion of the circulation reduced VO2 and VCO2 by 24 and 14.4%. That's expected because what we are doing when we occlude the circulation of the legs is like we are cutting out the leg metabolism from the rest of the body. So the legs cannot contribute to the overall oxygen uptake because they are not exchanging gas with the rest of the body because the circulation is occluded. And this reduction of VO2 and VCO2 was similar in males and females. This approach allows us to calculate the oxygen depth per kilogram of leg limb mass. And this value was exactly the same for male and females. During the occlusions with ischemia, the end tidal CO2 was reduced by almost 9%. And then tidal oxygen pressure was increased by 4.4%. This is likely due to the fact that ventilation is stimulated by the metaboreflex. And then there is more elimination of CO2 than observed in normal conditions. And this stimulation of ventilation is also rising the tidal O2 pressure above the level observed with free circulation. This is confirmed by the fact that during the occlusions, the ventilatory equivalent for oxygen and the ventilatory equivalent for CO2 and RER were increased, while tidal volume and heart rate were reduced, and these responses were similar in males and females. Overall, those results indicate that the respiratory gas exchange response to the occlusions was similar in males and females. And this is compatible with a similar metaboreflex response to post-exercise ischemia in males and females. We also look at brain oxygenation and pain perception. In this slide, we can see frontal lobe oxygenation during the experiments. And to our surprise, frontal lobe oxygenation was lower in the females than in the males during the first bout of exercise, and it remained lower during the consecutive bouts of exercise in females compared to males. And this effect was significant in the experiments performed with occlusion and open circulation, as we can see also here in the right side, where we have the overall effects in male and females. And also it's interesting to see how frontal lobe oxygenation is going down with repetition of both. It tends to be low, and this trend was statistically significant for the both performed with ischemic occlusion, as well for the both performed with free circulation. The muscle pain reported during the occlusions was similar in males and females. So to this point, I leave this open for discussion, and I would like to take the chance to acknowledge all the co-authors that participated in the experiment, the institutions that provide financial support to the studies, and especially, most importantly, to all the volunteers that contribute to make possible the study.
Excellent. Thank you very much for that. Again, I really like the way you've integrated numerous validated techniques to sort of answer your research questions. A very comprehensive study there. A little bit behind time, so we might actually jump over to Severine Lamon, who's going to take us through an infographic about how we can be or improve our study design to explore female physiology. And then after that, we'll take questions, and then we'll have the session, the networking session that we can also answer questions in as well.
Yeah, I just need to be able to share my screen, Lewan. I'm not able to at the moment.
I think that Becky might be able to help with that.
Yep, so let me share my other screen and go into presentation mode. Are you seeing what you should be seeing?
Yeah, I think so.
Yep.
Yeah, okay. So this is not a paper I wrote, but this is a paper I wish I had written. And I want to use that as an introduction for our infographic. So my name is Associate Professor Severine Lamon. I'm a physiologist working at Deakin University in Melbourne in Australia, and it's currently 9 a.m., I think, 8:30 a.m. And I've got an interest, I'm a muscle physiologist by training, and over the last 5 years, I've had a growing interest for how we actually report or do not report sex-specific differences in the field of muscle and exercise physiology. And a couple of weeks ago, this paper was published in eLife, and I wish I wrote this paper because it's a wonderful one. So I really encourage everyone to have a look. So basically, this paper looked at thousands of published articles available on PubMed and other databases, and classified them by discipline. And then they had a look at lots and lots of parameters regarding to whether they properly reported sex or gender specific differences or not in their papers. And one of the most important outcomes or findings that I found is this one. If you look at physiology, out of the paper that actually looked at sex specific differences, or at least looked at male and female cohorts, you can see that in the discipline in physiology, more than any other discipline except obviously reproduction, you can see that in nearly 50% of the cases, there are major differences between males and females. And in another 20% or 30% of the cases, there are minor differences between males and female. And only a small proportion of the paper who looked at male and female cohort reported no difference. So this is just to put things into context. As for my own journey, this is my mentor, Professor Douglas Paddon-Jones, and Doug sadly passed away in August this year, and he was someone who's been so important for my career. And he's the first person who a few years ago told me, Severine, will need to look at sex-specific differences in the response to exercise. And he's really the first who drew my attention on these really important sex-specific issues. And before his passing, he had secured already a few millions of funding, NIH funding, to actually conduct studies on sex-specific differences in muscle atrophy with aging and immobilization. And this idea has been with me for 5 years or more, but it's only in 2019 that my team and I published a paper in Sports Medicine, and it was around resistance training and skeletal muscle protein metabolism in females. And basically, we framed the paper as a framework. So basically, I started as a systematic review, but when we came to the end of the paper, we thought, oh, we really have to provide a framework here, because we've found all these great differences any solutions to researchers. So we first published the paper, and a few months later, we actually decided to create an infographic with the idea that an image speaks more than many words, and this is something we would be able to disseminate across the board. So this infographic that I'm going to present in the next 3 minutes or so is freely available at this address up here. So I'll post it in the chat after my talk, and it's really for everyone to use and disseminate, because we think that it's so important to consider sex-specific differences, especially in the area of exercise and muscle metabolism. And not many people do it, as Lewan said in introduction. So basically, our infographic is a framework to design female physiology research. Our original paper was based on muscle physiology or exercise physiology research, but the framework is applicable to everyone, and to everyone doing research in any area of physiology. And basically, it's basically just these are guidelines. It's not a recipe. They are just guidelines indicated what should be taken into account before designing a physiology study and accounting for possible sex-specific differences. So I believe that the first thing that people should do is to actually read the literature. And the first question they should ask is, okay, what is my primary outcome in my study? And is this primary outcome sex dependent, or says, no, go ahead, include your females, pull your males and females and run your study. But in most cases, the answer will be either yes or I don't know. And if the answer is I don't know, I strongly recommend to everyone to conduct a pilot study to just get a sense of whether whatever outcome you're interested in will be influenced by sex, and then, more complicated, by menstrual cycle. And once you've done that, you have, the moment you work with females, you have to work with a homogeneous samples. What does it mean? It means that unless males that are quite simple, and females are quite complicated, and we all know that. So you have all sorts of categories of females. You have females who are premenopausal. You have females that are eumenoric. You have females that are postmenopausal. You have females who are pregnant. And you really have to tease out and consider which factor you're actually interested in. Are you looking at a premenopausal period? Are you looking at a postmenopausal period? Because the hormonal profile will be dramatically different. So make sure you have a homogeneous sample to start with, and then look at the sample size that you can look at recruiting. If you're doing a muscle physiology study, as I do, and as our presenters today do, it's very likely that your sample size is going to be limited to a maximum of what, 20 per group maybe? If you are doing secondary data analysis or population-based studies, you may be able to work with a much larger sample size. So what does it mean? It means that both are okay, but you have to consider how you're going to control for these hormone level fluctuations across the menstrual cycle, and more generally drawing blood test, for example, at different times during your study. And for a large sample size, you could apply hormone level moderation just by running a statistical model where your hormone fluctuations are integrated as a moderator. And then comes the issue of what will we do with the menstrual cycle. So obviously, this is something that anyone interested in sex-specific outcomes in physiology has to consider. And again, our infographic is not a recipe. It's just a series of considerations. And what we ask people, or what we suggest people do, is to consider the menstrual cycle. And there's many ways to do that. You can ask women to keep a menstrual diary. You could draw blood tests and measure hormone levels. You could collect urine samples or use a temperature check. The best method is a combination of all of this, but people are limited by resources and time, and all these techniques can be used individually or in combination to actually help you design your study. And then comes the issue of contraception, and it's the same. You could decide to use oral contraceptive users, which will have a very standardized hormonal profile over the cycle, or you could choose to use non-oral contraceptive users, or both. It's all about controlling your cohort. So everything is possible. Again, this is just recommendations, but these things have to be taken into account in your study design. And finally, this leads to when should I actually test the females? And drawing on the cohort you've decided to test, the controls that you have applied, actually test your females. And when it comes to testing timing, this is a very rough depiction of the female menstrual cycle, where the ratio between estrogen and progesterone is quite stable during the first 7-10 days of the cycle. But basically, again, consult the literature and look at whether specific phases of the menstrual cycle should be either targeted or avoided. And avoided might be for reasons as simple as during ovulation phase, and don't quote me on that because I might say something completely wrong, females can produce more muscle strength than during the follicular phase. So ask yourself, what do I want to look at? Am I interested in the maximum force output that can be produced during the ovulation phase or across the cycle? And all these questions will inform the way you actually design your intervention. As always, crossover design is always more powerful than cross-sectional design. because you are just retesting the same woman over again in 2 different conditions. And ideally, all participants should be tested during the same phase of the menstrual cycle, if feasible. And I think that's the key point I'm going to finish with, if feasible. Female studies are so much more complicated than male studies. They cost more, they take more time, but they have to be done. So let's be thorough, and let's admit that we'll need more time and more resources, and consider all the things that I talked about today, and try to design the most possible thorough female specific or gender disaggregated studies in order to add to our body of knowledge of muscle and exercise physiology. Thank you very much.
Excellent. Thanks, Sev. Really appreciate that. It's just nice and straightforward, and I think at the very least it gets us all thinking about how we design our research, and that it is possible. So hopefully there's less excuses in the future. We have run out of time for questions, but what we will do is we have a networking session next or now, so we can ask as many questions as we want there and answer them, hopefully. So I think how that works is, Becky, do you stop the session and we join the new session, or does it just turn into the networking session?
Yeah, so if everyone just follows the link that I've dropped in the chat, I'll start that up and we can move over there.
Okay, all right, we'll head there now. Thanks everyone, I'll see you soon.